I'm Dr. Majumdar, CEO of PredxBio. At PredxBio, we are transforming drug development and discovery using spatial biology and spatial analytics. So very happy to be here. So there are two major problems in pharma. 90% of drugs don't make it, and the drugs that make it only work for small populations of patients. These are the two problems that we are attacking right now. At PredxBio, for the last 5 years, we have been accelerating discovery and optimizing clinical trials using explainable AI and unbiased spatial analytics. For a long time, it has been known that the tumor microenvironment is extremely heterogeneous, and it was thought to be very chaotic. One of the things that we discovered around 5 years ago was that there is some order to this chaos, and there is some organizing principle that determines the order that exists in this chaos. That order really is in the form of the microdomains that we discovered a few years back, which serve as guides to explaining the heterogeneity. And once we discover the microdomains, we are able to get into the network biology of cancer, which leads to novel targets and biomarkers. So, who are we? We are a company out of Pittsburgh, Pennsylvania, have been around for 5 years. Today, we have several customers, which you can see at the bottom of the page, who we serve with a proven ability to predict response to therapy with over 90% confidence. We're here to raise a Series A round, it's the Series A2. We already raised Series A1, which was for $2 million, and we oversubscribed heavily already on that. Today, we are here to raise Series A2. It's a participation preferred stock with a $22 million pre-money valuation. Huge momentum for us the last 6 months or so, when we have got a number of pharma customers to come in: Bristol Myers Squibb, Merck, Genmab, AstraZeneca, and 20-plus peer-reviewed publications in journals like Nature, Communication, Cell Reports, etc. 10-plus patents in the last few years granted. A number of these are US patents and some international. And Stan Marks is the chairman of our board. Stan Marks is the chairman also of the Hillman Cancer Center in Pittsburgh, Pennsylvania. So what's a big problem that we are tackling here? For the last 10 years, we have known that immunotherapy clinical trials have been growing tremendously, 250% increase over the last few years. The thing that has remained constant is the response to therapy. 17% of patients respond to immunotherapy at advanced stage. The rest do not really respond. As a result of this, what happens in pharma, of course, the patients are in agony, but the pharma, after spending $2 billion, 10 years, only 10% of their drugs are successful in immunotherapy at this point of time. This is largely because, as I said, about the heterogeneity in the tumor microenvironment. And biology in cancer is immensely complex. This is what is extremely difficult to get into the underpinnings of, and that's what we focused on. So 5 years ago, we asked the question, what if you could identify subpopulations of patients who would respond to immunotherapy with over 90% confidence and radically improve clinical trials and the discovery process? This is what we have been able to deliver, and we are very proud to say that over the last few years, we have been consistently delivering this kind of accuracy in terms of predictive response to immunotherapy versus the PD-L1-based tests, which are the standard of care in the market. We have been doing this consistently from biopsy tissues, which are labeled with X number of biomarkers. It could be anywhere from 2 to 3 to 5 to 100 biomarkers. And we are able to leverage spatial biology to look at the entire tumor microenvironment in the tissue, with the complexity of the tumor that you have with stromal cells, tumor cells, immune cells, T cells, B cells, all tweeting at each other, communicating with each other. We capture those on the basis of the proteomic biomarkers, and we also capture these microdomains, which are constellations of cells coming together in an identical spatial orientation. And this is going to be our secret sauce to connect that to the outcomes. Once we get the microdomains, we are able to leverage what we call explainable AI, which is the next level of AI over deep learning, machine learning, etc., which is there in 99% of the different AI algorithms that you see from different companies. With explainable AI, we're able to discover and reason about cause and effect. And that's going to be our secret sauce to leverage that and expose the network biology. This is our platform. We can ingest data from proteomics, transcriptomics, simple bright field pathology. And within a fraction of a second, we are able to phenotype every single cell that is there on the tissue using this recursive tree and leaf algorithm on the right-hand side that you can see. Once the cells are phenotyped, we know who's who, tumor cells, stromal cells, macrophages, etc. And once we know who's who and who's located where, we are able to generate these microdomains, which are constellations of cells coming together in an identical spatial orientation. As I said, these microdomains are critical in terms of some of these could be tumor promoting, some of these could be tumor restraining. And once we discover these microdomains, we can correlate that to the network biology of cancer. So we have published all of this over the last few years in a number of different journals, from Nature Communication to Cell Reports to Discovery, etc., marquee journals across the board. And we have also patented this technology, 10 plus patents, as I said. So let me show you some use cases in the last few minutes that we have, from discovery to clinical trials to translation. One of the things that we do is we not only predict all of this with a very high degree of accuracy in terms of response to therapy, but we also expose why. Why is this happening? Why is this subgroup of patient responding versus why is this subgroup not responding? Because we get to the pathways of cancer. And that has kind of been our secret sauce over the last few years. These are some of our customers, some of the projects that we are working on. Genmab, optimizing immunotherapy clinical trials, Bristol Myers Squibb's, predicting response to combination of immunotherapies, etc. Let's get into some of the nitty-gritty. This is for a company called Genmab that we have worked on for the last couple of years, disease, diffuse large B-cell lymphoma. The challenge was to identify responders versus non-responders in immunotherapy clinical trials. We're able to do that with 93.3% accuracy compared to where Genmab was, basically 0.60 AUROC. Dramatic improvement over the last 6 months that we've been working on with them. And we have shown this consistently across different trials from biopsy tissues labeled with 8 biomarkers. And as you can see now, Genmab has put us across their entire range of clinical trials that are going on lymphoma, as well as some of the other solid cancers. And we are working our total pipeline from phenotyping to microdomain discovery to network biology with them. This is one of our very happy customers. Chung-Wing Lee talks about how we helped them in phase 1 and 2, and now they're going into phase 3 in terms of predicting the response to therapy. This is for colorectal cancer. The challenge was to predict the progression of colorectal cancer with a certain degree of accuracy. We're able to do that with 93% accuracy in terms of a 400-plus patient cohort, predicting their 5-year risk of recurrence, compared to some of the other players in the market, Akoya, Exact Sciences, etc., who are in the 60 to 75% range in terms of accuracy. This is for BMS, revealing the mechanistic difference in response to combination of 2 immunotherapies. We're able to really explain why the combination works way better than a single checkpoint inhibitor. And again, Sarah Hersey, one of our BMS VPs, very happy with what we have done in terms of exposing the mechanistic underpinnings of 2 checkpoint inhibitors versus 1. This is our pipeline at the moment. These are customers on the right-hand side. The market is easily over $100 billion for what we are doing right now across various cancers and across different therapies coming up, including CAR-T and cancer vaccines. This is our revenue profile. We're going to make $4 million this year and rapidly increase. Some of the offerings that we have right now, going from opportunistic services all the way to spatial concierge services, as well as platinum service with microdomains and network biology. Come to our booth, take a look at this amazing view of the tumor microenvironment with our spatial computing with the Apple Vision Pro. We don't really have, at this point, a direct competitor, and we can talk more about it when we meet. This is our exit strategy. We are going to exit at the end of 26, 27. Very good valuation at this point to come in and get a huge return. Some of our target acquisition partners, the ones you could expect from pharma, as well as some of the data companies. This is the team. I come with around 25 years of experience from GE Healthcare, IBM, Exact Sciences, etc. Chakra, who's sitting here, comes from Jeff Hinton's lab, the guru of AI. And I hope that you come in and have more conversations with us about investment, a de-risk platform, great market demand, good customer traction, a world-class team that we've assembled of oncologists, of physicists, of computer scientists, and AI folks together. And we hope to talk to you about investment in Series A2. Thank you very much.